Great. Howdy, my name is Rob Thoms. I'm with Stillwater Sciences, and today you're going to get your GED in GDEs. My dad was a general education development teacher. I always thought he was a lousy teacher, so I'm going to try and do better. What's the point of generations? It's okay. I'm going to hit you from the start with Mini Wichoni, water is life. Alternatively translated here as groundwater is essential. Groundwater is essential to California's agriculture, urban uses, flora, fauna, and as Kristen highlighted in her Drake's Bed example, carbon sequestration also. The problem is groundwater pumping and land conversion in California have created a suite of environmental problems, including land subsidence, increased costs for newer, deeper wells, lower base flow for rivers, which affects recreation and aquatic communities, and ecological impacts to vegetation communities. For example, less than 6% of the historical riparian vegetation and wetlands remain today in California. So the solution, at least for now, State Groundwater Management Act. This was landmark legislation enacted in 2014. Unfortunately, it set the groundwater levels in 2015, which was, year 6 of a 7-year drought. But it established agencies to sustainably manage groundwater resources. It requires that all groundwater basins create a plan for sustainable groundwater withdrawals within 20 years. And it states that groundwater-dependent ecosystems be identified and impacts considered when setting sustainable management criteria, particularly where species and habitats are protected by federal, state, or local legislations. So, GDEs, groundwater-dependent ecosystems, are ecological communities, plants, animals, natural communities, and they can be found in a variety of different environments. Seeps and springs, groundwater-dependent wetlands, meadows, fens, and river corridors enriched by groundwater or groundwater surface water exchange. Groundwater-dependent ecosystems include nearly 400 mapped natural communities, over half of which are designated as sensitive natural communities, and many are habitat for special status plants, wildlife, and fish species. As you can see in the healthy system, we've got riparian vegetation, we've got some salmon in there, we've got mammals around. So some interpretations of GDEs. Steelhead often benefit from groundwater inputs to maintain sufficiently low water temperatures. Not just water volume, but a cool water temperature requires groundwater. And in the unhealthy system, you can see that pumping alters the groundwater elevation, and the water table declines below the rooting depth of some of the riparian vegetation. That's a direct impact. And say the southwestern willow flycatcher that requires that riparian vegetation gets an indirect effect from groundwater pumping. So putting this into practice, Stillwater Sciences has incorporated groundwater-dependent ecosystems into analysis for some groundwater sustainability plans throughout the state. And to effectively protect ecological resources through the Groundwater Management Act, the following data are crucial. High-resolution depth-to-groundwater modeling data, including information on shallow groundwater, high-quality vegetation mapping data, rooting depths of plant species, and understanding groundwater-surface water interactions. The secondary problem is these data aren't always of sufficient quality to identify GDEs for inclusion in sustainability plans. So groundwater depth data can be scarce. There are a few wells that record that depth, and many of them are private. Vegetation mapping in many areas of the state is still too coarse. We're cheering you on, VegCamp. And rooting depths may be inexact or unknown, or not have a very large sample size. So you can see there are many research opportunities here. Clearly this work is interdisciplinary. Thank you for letting me get up here and talk about hydrology and geomorphology and fish and wildlife during a botanical conference. But everything in nature is connected, and this is another mechanism that we can use to protect some of our natural ecosystems in California. And I've got a couple coworkers, Megan and Carly, who have a poster during the poster session, so you can come see us then. Thanks.
Good afternoon. My name is Rob Irwin, and I'm here to talk about a website, which I know you guys all love, so let's get right into it. And the website's called the California Species Mapper. A little bit pretentious, but hey, that's what it is, and nobody took the name, so I took it. Okay, so it's just a wonderful world we live in. There's so much data out there, and I'm sure you guys have to deal with it. And one thing I'm interested in is biogeography, and I need to know like which species are in an area for my SQL and NEPA work. And I know a lot of people do, so I thought, hey, there's some cool sources out there that I've been using. They're a little clunky sometimes, and there's several of them, so it's hard to kind of get data from this site and that site and compile it. One, if you guys don't know, there's this website called Bison. It's from the U.S. Geological Survey, and it is fantastic. And you can search for a species and hit go and hit points and see a bunch of data from hundreds and hundreds of sources, like museums, universities, California Consortium of Herbaria posts their data on bison. It's a rich data source, and it covers the entire United States. Let's see if my green eyes are really. So another great website, online database, if you guys don't know, is GDEF. And I never say the full name, but it's the Global Biodiversity Information Facility, and it's got over 1.3 billion observation points for different taxa. It's a global data set. And bison is feeding into here and feeding back, and it's fed into a lot of state, local, international databases. It's a great repository for data. Unfortunately, it doesn't have very many mapping facilities. It's kind of clunky. So, another great resource, if you guys haven't heard about, is iNaturalist, and this is kind of a community-driven effort, and anybody can submit observational data, and it's reviewed within the community. And if two people agree on a species, it becomes part of a research-grade data point. And it's growing exponentially, and this is going to start swamping a lot of other data sets, and there's taxa that pop up in places that you'd say, hey, this doesn't occur in any other, Cal Florida doesn't have it. But somebody found it, and they put it on iNaturalist, so it's a good source to check sometimes. So here's my website, which is basically trying to grab these data sources, so GDEF, Bison, iNaturalist, and make it one interface so you can search and see the points quickly on a map, so you can do some cartography with it and look at other layers. So here's a run where I'm searching for Calochorus, and you can see it's coming up with 6 species around Lake Tahoe, and you can see there's nothing in Nevada because it's a California species mapper, and so I'm just saying, rest of the country, doesn't matter. So, some things you can do is you can hit this button right here and copy the table and paste it in Excel, so it's a quick way to get it tabular data. You can do fun cartography stuff with it, so I changed the base layer so it's elevation, stretch gradient, and I changed the symbology for some of the species. So there's a legend, and there's tools in there, and you can click a point and then see the data that comes out of it. So this data set is from, I don't know, I should move on here. So another view of it, you can measure points. Oh, so here I clicked on the view table of this downloaded data set, and I can see all of the records that came through from bison. I think this is Plante, so I just went and said Plante, and I got all the plants around Bidwell Park, and I can copy that and put it in Excel if I want. I can click a row and zoom to a point. So here I can symbolize by species. So I downloaded a genus, and I said, okay, draw it as different colors. I can drag and drop other data. So this is a soils data from Surgo, and I just got a KML, or I think it was a KML, drag it on. Oh, I think that's my time. So anyway, thanks for listening, guys, and I hope this tool is useful to somebody. So good.
Okay. All right, so I'm Adam Schneider. I'm going to be talking a little bit about some research I've been doing with mistletoes. And you may have heard me here from previous conferences talking about my other work in parasitic plants. I did my PhD in Berkeley, but now I'm at Hendrix College in Central Arkansas. And one thing I just kind of want to mention is that I'm still working on that group, Aphyllon, formerly Orobanche. And there's a student of mine is giving me a poster on it's going to be a newly redescribed species. So something that's widespread that we haven't really recognized as unique before tomorrow morning. So I hope you check that out. One of the cool things about, for me, studying parasitic plants is that it's evolved independent. This idea of heterotrophy or losing your ability to do photosynthesis has happened a whole bunch of times evolutionarily. And so we can look at these different groups of organisms and see these generalities from an evolutionary perspective, or test generalities. So on the left here, you can see kind of 3 different examples of cool non-photosynthetic parasitic plants that are here in California, and there are others as well. So one of the things I found from my PhD work is that there are these cryptic host-specific species in Aphyllon. And long story short, I got connected with some people who are working on dwarf mistletoes and became interested in this controversy that's arisen in the last maybe 10 or 15 years about how many species of dwarf mistletoes are actually in California. And if there are lots of species, you might say that there's lots of mostly host-specific species. And so that's kind of the traditional view. Hawksworth and Weaves, who wrote a really important monograph, considered 12 different species of dwarf mistletoes here in California. Some people still hold that view. You can tell these apart using a combination of different morphological traits as well as host preferences. However, in 2003, Dan Nickrent and colleagues published the first molecular phylogeny of this group. And that molecular phylogeny was made using ITS and a chloroplast spacer gene. And they found really no support for a lot of these different species. They couldn't tell them apart using these 2 genes. And so that's where you get the current eFlora treatment, where there's only 3 species here in California. Most of the taxonomic diversity has kind of been collapsed into this Arceuthobium campylopodon. Some people treat these at the subspecific level. You can get into those kind of taxonomic arguments. But what I wanted to do is to try to see if we can use next generation sequencing techniques to better resolve this phylogeny in hopes of adding some additional data that taxonomists who are more of an expert in these plants than I am can use to try to fortify, or maybe we can come to some sort of consensus about the diversity here. So I used a technique called genome skimming. Basically, this is a way of sequencing high copy regions throughout the genome. So chloroplast sequences and ITS, ETS pretty cheaply. So I did that in order to, and in addition, sampled additional taxonomic samples, and also wanted to properly root the tree to try to figure out what the relationship was between our North American taxa and the European Arceuthobium. So long story short, I don't have time to get into all of this. I'm hoping to publish it sometime this year. So just keep an eye out for it, and there'll be some interesting results. But just as a little teaser, we found that chloroplast and nuclear data are relatively congruent in terms of the phylogenetic patterns across the genus level. I know you can't read the details here, but again, you can hopefully look through this paper and dig into that if you're interested in these dwarf mistletoes. But I want to leave you with kind of 3 big takeaways. First, we still don't find resolution in that section campylopodon, even with about 15,000 nucleotides from the chloroplast and nuclear genome. And there are reasons why we shouldn't necessarily say that these aren't distinct, and I can talk about that later individually if you're interested in. But the solution of just throwing more data at this problem, is not going to resolve these if they are indeed different. Second, strong support for sister Old World and New World clades. And finally, we see some good section level taxonomy.
Hi everyone, I'm Rochelle Bull. I'm an environmental scientist with the California Department of Fish and Wildlife's Vegetation Classification and Mapping Program, otherwise known as VegCamp. My purpose in speaking with you today is basically to either remind you or tell you for the first time what we do and where to find our data. So VegCamp, in conjunction with the California Native Plant Society's vegetation program, develops and maintains California's expression of the National Vegetation Classification System. We also develop the state's suggested practices for vegetation sampling, vegetation classification analysis, and vegetation mapping, with a goal of completing a fine-scale statewide vegetation classification and map. All of this is to say that we have a lot of publicly available vegetation-related data that we want you all to use. So this is how you access all of it. Let's start with our classification. It is quantitative, meaning data-driven, and it's hierarchical, meaning that it can be viewed and used at multiple levels depending on your goals. You might recall the book version of the Manual of California Vegetation up there in the right-hand corner. We now have an online version that is updated regularly. You can find it online through links from the CNPS or the VegCamp website. You can see, here's an example of an alliance level information through the NCV online. There's associations listed for each alliance at the bottom with references from where they were described. There's a lot of information for each alliance, but I want you to know mostly that, you can't even read this, but over here on the right-hand side, there is global and state rarity ranks for each alliance, which brings me to my next point that most people are unaware of, that yes, community types are given rarity ranks. We use the NatureServe ranking calculator. You can download the calculator and read more about it from their website. This is the manual how to use it. We rank communities at the global and state level from 1 to 5, 1 being the rarest. Communities that have ranks between 1 and 3 are considered sensitive natural communities. These rarity ranks are especially important for environmental review and determining project impacts to sensitive natural communities, among other purposes. You can read about our suggested protocol for how to determine impacts to sensitive natural communities from this document here that can be found through our website. Moving on to our maps. They're available for download and viewing on BIOS. If you just search BIOS CDFW in Google, it'll get you here. This is what the viewer looks like. Search vegetation at the top, circled in red, and it'll show you a list of all our vegetation maps. This here is a map of our maps that you can view from BIOS as well. From this map, you can see what each map is called, and a little bit about each one, who mapped it, the year that it was mapped, its status, if it's in progress or complete, stuff like that. You can add individual maps to this viewer, and then you can view the metadata, which will redirect you to our website for the full report. And from BIOS, you can also download the data and to be used however you want to. So, and last, well, you can also see that, so like I said, this is the map of all our maps that meet our state standard. You can see that the state is not complete. We're 60% done, I suppose. You can always feel free to contact us if you have questions. There's no data in your area, or also if you'd like to contribute data to us. But speaking of contacting us, I'd also like, the last thing I'd like to do is point you to our VegCamp website that I keep mentioning. You can just Google VegCamp CDFW and it'll come here. You can find links to everything I've already talked about, plus much more. You can access all the reports, sampling protocols, data sheets, our standards, and even a vegetation mapping geodatabase template you can download from here. You can download a full natural communities list as well as a list of sensitive natural communities, all with rarity ranks and all sorts of other things. And then, of course, there's our contact information. Can't find something, have any questions, or have data that you want to share with us, please do not hesitate to contact us. Thank you.
Hi, I'm Tim. I'm up here because I'm looking for people to chat with at the reception after this. I'm interested in setting up some long-term monitoring programs in association with the course that I teach, California Ecology and Conservation, and I'm looking for ideas and collaborators and that sort of thing. So before I get to the long-term monitoring project, a little bit about the course. So the course I teach is called California Ecology and Conservation. It's a 50-day field course run through the University of California. It's open to students from all 9 campuses across the UC system. It runs spring, summer, and fall. We take students to across the state, even Southern California, but everywhere from Anza Borrego the whole way up to Angelo Reserve up in North Coast Ranges. We hit most of these sites about once a year, and if you're interested in knowing anything more about the course, follow us on Insta, and you can also check out our website at ucnrs.org/teaching/CEC. I'll repeat that again at the end. So most of the time on the course, the students are working on their own independent projects. We spend about 10 days at the site, and most of that time is working on those projects. The first couple days, though, the students are getting their feet wet there, and they're studying the natural history and reading some of the primary literature about the place. And during that time, we have some time for them to work on other projects. So it's basically an opportunity to set out some long-term data collection for free. So we started to do this already. This is Blue Oak Ranch Reserve, kind of near San Jose. If you haven't been there, get there. It's really cool. It's a Berkeley-run reserve. And we started out an oak monitoring protocol. We planted about 1,000 valley oak seedlings in a big macro plot. Valley oak has some recruitment problems. There's a lot of ideas for why that might be. One of those is that there's competition with non-native grasses. So we mowed half of a plot, and we wanted to see how well the oak seedlings did in the aftermath of just one mowing treatment put out a couple weeks after we planted the acorns. We collected data the past couple classes, and just one result, it looks like plants or little seedlings in the unmowed treatment were taller than plants in the mowed treatment. I have some ideas about why. If you're an oak head, I'd love to chat with you more about that. But the main point that I'm trying to make here is that with just a couple afternoons with my class, I was able to get some high-quality data, and I want to replicate that across a lot of the different reserves that we're going to. So to give you a little bit more information about the parameters, we hit most of these sites once a year. There's a couple exceptions to that. But we tend to get there at the same time every year, so we can repeat this. And I want to do this job forever, so hopefully for decades to come. We have 27 students that take the course, and I can give about 4 hours before the students start to get tired and need to eat and things like that. But 27 times 4 hours is actually a lot of person hours to throw at something once every year. So I have some ideas, and this is by no means an exhaustive list of some stuff that might work really well for this sort of setup. I want to set up some long-term phenology plots at a place. Maybe spring would be a good time for that, looking at the timing of flowering or fruiting in a particularly interesting community. I think studying community shifts, say in the aftermath of a fire, might be really interesting and worthwhile. Putting out some permanent plots for something like that. I'm a pollination biologist by training, so obviously floral community type, visitor community type stuff might be interesting. And then herbivory studies, so looking at gall-forming insects or leaf-mining insects on a particular species of interest. But I would love to pick you guys' collective brains about some other potential things that I could do on the course. And so please talk to me. Also, that's the right email. The email in the program is wrong, so use that one. Thanks.
Oh, good. That one's actually mine. All right. Hello, everyone. My name is Katie Pearson, here from Cal Poly State University, and I'm here to tell you about an urgent need in the botanical community, a way that you can help advance California botany from the comfort of your own home, library, office, or school. Well, what's in it for you? Let me ask you a question. Does the winter's cold have you stuck inside, missing your favorite plants, dreaming of spring botanizing? Well, you can see your favorite plants every day as high-resolution images of herbarium specimens on the new data portal cch2.org. CCH2 is an extension of the Consortium of California Herbaria database, which is now known as CCH1. And CCH1 contains all vascular plants, only vascular plants collected in the state of California, while CCH2 includes all available data from herbarium specimens from herbaria that are in California, regardless of whether those specimens or where those specimens were collected. As a result, CCH2 is an online database of over 2.4 million herbarium specimen records from 54 herbaria, including the Chico State Herbarium, and we're trying to include all California herbaria in this database eventually. Hundreds of thousands of these records include a high-resolution specimen image, and that's thanks in part to the California Phynology Network, which is a collaboration of 22 California herbaria funded by the National Science Foundation to image, there's a map of us, to image nearly 1 million specimens by 2022. So to date, we have created about 250,000 herbarium specimen images, which are available on cch2.org for scientific research, especially about how these plants are responding to climate change, particularly in terms of their phenology, so how their flowering and fruiting times are changing. However, images alone are not sufficient to empower this type of research. We need to digitally capture the specimen data from these specimens and make them into these digital data. And that's where we need help. One second. There we go. We're getting there. All right. So we have put thousands of our images on this website called notesfromnature.org. And here, any member of the public can come on and look at these high-quality images and learn how to transcribe information from these data labels into provided text fields. I know the quality is really bad. It's just generally how it goes. And so these data are then quality checked and then imported directly into CCH2, where they're instantly available for scientific research, education, and even your own personal use. So many herbaria in California have never previously been digitized. And so with Notes from Nature and help from people like you, scientists, citizen scientists, we can make these dark data come to light. Right now we have expeditions from the Fresno State Herbarium, from Cal State LA and Long Beach State, and UC Irvine, with many expeditions still to come. Who might finish? Who's going to finish first? Well, yeah, that could be up to you. If you like UC Irvine, you could be like, no, they're gonna pull ahead. So, herbarium specimen data form the basis of our understanding of where plants occur and how they're responding to climate change. But we need your help to make these data available from the hundreds of beautiful specimen images that we're making available on cch2.org through the California Phynology Network. So, on behalf of the California Phynology Network and California Herbaria, I invite you, I urge you, to go to notesfromnature.org today and spread the word and help these California Herbaria in our transcription and digitization goals. So I have a bunch of flyers that can help you understand how to get to this website and use this website. They're over at the Friends of Chico State Herbarium booth. I'll also be carrying them around, and I can answer any questions you might have in the reception. Thank you for your attention.
Hi, I'm Cecile Showhead with the Medford District BLM in Oregon. I'm here to let Northern California wildland botanists know to be on the lookout for shiny geranium. I first encountered shiny geranium in a conifer hardwood forest in the lower Applegate watershed in Oregon. Alarmed would be sugarcoating my reaction. This isn't the Willamette Valley or the Olympics. The Klamath-Siskiyou bioregion doesn't have naturalized annual geraniums which dominate the herbaceous layer in our closed canopy native forests. But now we do. Shiny geranium or Geranium lucidum is an early detection, rapid response, annual, aggressive, invasive, new to the Klamath-Siskiyou Mountains in southwestern Oregon. It moves directly into forest habitats, spreads rapidly, replacing native herbaceous plants, annual and perennial forbs, and grasses. It has a real significant capacity to naturalize. So currently, we know of about 125 acres, 120 are in forested habitats, the rest are roadside, and in the floodplain of the Rogue River, unfortunately. And we expect to find more. So these three photos depict shiny geranium sites in our forested habitats. And, okay, so the photo on the left, right here, is a gap, an opening in the forest. And everything on the forest floor that you can see is shiny geranium. There's likely tens of thousands of seedlings. So I just want you to note that these photos were taken in the winter, in late December. Shiny geranium is a winter annual. It germinates in the winter. The other two photos are depicting just canopy coverage. Okay, so a little bit about the biology. So shiny geranium, a prolific cedar, as annuals are. Copious numbers of plants, exponential numbers of plants. It's a winter annual, which means it germinates in the fall or the winter when the natives have died back. Shiny geranium then occupies their space, and in the spring, the natives are out-competed. So, some known habitats in the Klamath-Siskiyou ecoregion. So we've talked about forest types, openings to 80% canopy cover. Shiny geranium loves intermittent drainages under canopy, and it spreads rapidly, just scootling seeds downstream. Lots of microhabitats, varied aspects, moisture gradients, soil types, and slopes, varied slopes. It has a patchy distribution, sometimes a strong monoculture, sometimes dominant, sometimes less frequent, hiding amongst natives. So we also know it's in riparian areas and in oak woodlands. So now, let's talk a little bit about identification. In my area, Geranium lucidum, or shiny geranium, can be confused with Geranium male. And so, shiny geranium is 5-petaled, and the petals are unlobed, so that the flower appears to have 5 petals. The leaves are round with 5 to 7 lobes, and the sepals are inflated or keeled. So some of the tissue comes out to a sharp edge. The stems are often reddish, and that is usually in late spring and summer, and the plant in general is not hairy, which is a really good vegetative, like winter and fall characteristic. So, Geranium male, on the other hand, has also 5 petals. They're deeply lobed, so they appear to be as 10 petals. And the plant has soft, downy hairs on the stem, on the calyx, on the petiole, and somewhat on the leaves. Also note that the calyx is not inflated and it's not keeled. So, which geranium is geranium number 3? Awesome. How about 4? Yeah. All right. Have a great afternoon.